I am happy to answer any questions. And the rest of you should feel free to flee at your yes?
So I was curious, it's obviously not possible to measure gene expression in real time in the human brain, but you mentioned that, or not yet possible, right? You mentioned that there are bioinformatic ways, there's tissue-specific expression, and you said that there were bioinformatic ways to make some indirect inferences about that. So my first part of my question was, what do we know about expression in the brain and maybe region-specific expression, if anything? And the second part of my question is about time course. You mentioned a period of several months. You also mentioned earlier that sleep deprivation changes gene regulation. I was wondering if there's any evidence that of more sort of like the Incredible Hulk view of gene expression, where I get mad, say, there might be genes turned on during a very short time course of emotion, or let's say while you're having sex or something like that.
Yeah.
An event that might last relatively briefly, is there any evidence for expression at that time scale?
Yeah, there's two answers to that. So one, the intrinsic on time for turning a gene on is probably, in the tens of minutes. So it takes about 5 minutes to get the transcription factor on the DNA at peak level. It takes about 20 to 40 minutes or something to get RNA transcription up to the level that it needs to be to have a discernible phenotypic effect even at the cellular level. And then the appearance of protein follows pretty quickly after that. So for protein studies, we usually look about 3 to 6 hours after sending a pulse. That's when we start to see peaks from the first round of expression. So there's probably not at the time scale of the Incredible Hulk, where he blows up out of the phone booth in 30 seconds or something like that. But the first round of gene expression can come on board within several tens of minutes after a pulsatile signal is sent. So after that, it becomes somewhat complicated because it looks increasingly like much of gene regulation is actually a secondary consequence of what happens in the first round. There's certain, if you will, first line of defense responses that are programmed initially, but after that, the system becomes sort of very feedback sensitive, but also open to experience from the environment. So they tend to become somewhat unstable systems, and so the big take-home answer to your question is really, when you're going to change somebody into the Incredible Hulk, is not by having a big response for a short period of time, a big pulse. It's by having a smaller, arguably, or similar, experience that's protracted over time, that happens day in and day out. So this, makes me think of, I've got Shelley Taylor here. She's going to probably tell me all the ways in which this generalization is slightly wrong, or maybe hugely wrong. But my read of the health psych literature in general, looking at socio-environmental risk factors and sort of biological responses, is that acute one-time stressors make you miserable, but they don't tend to be the biggest predominant sort of epidemiologic influences on health outcomes. What makes a bigger difference there, in general, are smaller, more protracted, let's say less visible or less experientially salient, more protracted experiences. They're conditions of daily life that actually often recede out of our experience. So the human perceptual system is very good at picking up changes. What we don't notice is the bad things that are there every day. We habituate to them really quickly. That's why things like socioeconomic status seem to burn in so strongly. That's also why things like worldview and personality make such a big influence empirically, as they sit there day in and day out, pecking away at the decisions that your system makes. Another way of thinking about this is in terms of the kinetics of remodeling or fluidity. So remember I said you're replacing 1 to 2% of you every day. Now that's independent of any kind of separate induced pulsatile gene expression response, but there is this kind of input to your basal gene expression that's happening at the level of maybe 1 to 2% replacement per day. You have a really bad day, you're sending a really big message to 1 to 2% of this self-regeneration process. If you have 100 bad days, you've suddenly had a chance to color in some sense, kind of an entire replacement cycle. Now, that math isn't strictly correct, but basically, the scenario that small protracted differences for a long period of time may be both more invisible to us and ultimately more influential, because they're the things that are structuring this ongoing regime. So that when you get to the end of the first round of gene expression, and the system becomes then very unstable and sort of environmentally sensitive, that's when having another bad day says, yeah, make a lot more IL-6. You have bad days after bad days after bad days. That tends to be where we get this. What do you think? Reasonably close?
Yeah, I would agree with that. I think grinding chronic low-level stress of which people are not aware because they've incorporated it into their expectations for their lives is what wears people down after these kinds of processes. Well, I have your attention. May I ask my question?
Sure. I haven't forgotten, though, that you had another question, so I'll come back to you in a second.
I wondered to what extent you think the causal direction goes at least in part in the opposite direction. You've talked about the social environment being the causal agent, but there are data, for example, on hostility that suggests that it may be at least in part a reflection of underlying genetically based differences in reactivity, and there's data suggesting that loneliness has a very large depressive component in depression itself, or both loneliness and depression are associated with social withdrawal, which is in essence social isolation. So there are at least some signs that the direction of causality can work the other way around, that changes in your inflammatory responses can influence your social environment. So
Yeah.
you think that's a big player, medium player, or a small player?
Well, I mean, the first part I can agree with 100%. These are definitely reciprocal determining systems, and probably what's most important. I mean, at some point it becomes hard to carve up how much is due to this versus that if they both run in a cycle, because you wouldn't have this much from A if B weren't voting for more in this kind of thing. So the bigger questions become, when are the circuits self-propagating and when are they self-correcting? And I think you hit on the best example of the self-propagating system, which is the you can start either with genetic liability for making more inflammation than influencing your brain and sociality, or you can start with adversity coming in from the environment, making your brain, essentially more, sort of pro-inflammatory in the signals that it's sending to the genome. In the circuit, it turns out it doesn't matter that much which part you start with, because they're both going to keep going forward in the same direction.
And the animal studies address that.
Yeah, exactly. So the animal studies clearly say that both are true. In other words, you can manipulate inflammation and get changes in social behavior. You can manipulate social behavior, get changes in inflammation or antiviral responses. And they're sadly in this kind of self-propagating, sort of circuit. Now, that's not necessarily going to be true for all contexts and gene systems, but certainly for that one, it's that's probably our best evidenced example. So yeah, I totally agree. And then that question then, sort of raises the next, kind of the applied intervention question of, where should you intervene in these systems? I mean, my response to most people trying to, I've got Susan Lutgendorf running around trying to do cognitive behavioral therapy or, that kind of thing with her ovarian cancer patients. And if, cognitive behavioral interventions were really terrifically successful at changing people's worldviews, so much so that they now thought the world wasn't a threatening place, even though they've got ovarian cancer and will certainly die within the next 2 years. That's a hard one to ask, for instance, behavioral interventions to affect. So in scenarios like that, we might be inclined to say, give them a drug that just cuts the sympathetic nervous system off at the legs, so that at least the difficult trajectory that they're contending with isn't going to be driven even more strongly by that side of the circuit. But at the same time, to everybody who's running around trying to cure cancer or inflammatory related diseases without attending to these, sort of native propagators from the social world, I think that's equally sort of doomed unless you have a very good, specific, completely, downstream drug, for instance, targeting just inflammation, which, generally we don't have right now. So I think the thing that's going to work best are the things that, it sounds like I'm sullying psychologists, which I don't know, arguably maybe I am, but I think what's probably going to work best are kitchen sink interventions. I think Barbara Anderson has it right. Do everything. Sort out what the crucial ingredient is later. If you've got a circuit the best thing for stopping a circuit is intervening at as many points, I think, in the system as possible. So that's what I take away from the circuit notion, is that, instead of trying to intervene here or there, it just gives you more opportunities for intervening in the same system. Yes?
A wonderful talk, really very exciting and kind of well transforming. I was really interested to come up there.
It was a long journey, but I made it.
Yeah. Well, it's a different world down there. A quick speculation and a question, because I know we're running out of time. But the quick speculation is that I wonder if this has to do with the massive epidemics you get with culture contact in the world and that sort of thing. Whether it's not just that people are exposed to new viruses and so forth, but that's compounded by social stress and it makes people more vulnerable to those viruses. And so you get these epidemics that wipe out 50, 60, 70% of the population. It may not be just that they don't have the innate immunity to those things, but they're compounding exposure to new viral agents with the stress.
With a poor response, yeah.
The other question is, and maybe it's not quite fair because it's not exactly what you're working on, but in what ways does gene activation and transcription in response to social experience produce different kinds of social behaviors? You've gone from the social to the immunology, but you would think that social experience, through the same kinds of general mechanisms, would lead you to respond differently and not just respond differently, but to initiate different actions and so forth, based on what you or maybe you and your mother experienced. And besides stress reactivity, do we know anything more specific about that, or?
In general, that possibility is kind of what I was hand-waving at with the rat prefrontal cortex, and we can see the same thing in the amygdala and a couple other, sort of general systems, or at least a chunk of tissue that includes the amygdala. Rat amygdala is pretty small. What exactly do those gene expression changes in, let's say, the prefrontal cortex portend for social behavior? We don't actually know that. Going from genes to these kinds of phenotypes has been extraordinarily hard, much more difficult than I think any geneticist ever anticipated. And yet we know that there's profound genetic structuring of these things. It's just happening at an ensemble of many hundreds, if not thousands, of genes, probably not at the level of the individual genes, at least typically. That's why that IL-6 example was so atypical. There's very few genes that have that much play over a phenotype. So at this point, I can't tell you empirically, we know a lot about this gene going to that behavior, with the possible exception of, let's say, the sort of exactly the world you were talking about, the neuroendocrine responsiveness world. So, for instance, Michael Meany's studies showing changes in the glucocorticoid receptors expression in the hippocampus as a function of early life social conditions, then changing essentially the feedback sensitivity of that system, and presumably, rendering the adolescent and adult organism essentially more vulnerable to stress and less sort of stable in its response, and then presumably as a function of that performing worse in its social relationships. So there's a reasonably tight heuristic argument you can make, but as far as I'm aware, there's nobody who's really charted every step of that. I guess the other literature I should think more about before really, probably the other literature you should think more about, since I'm not sure I can even play this one out to the end, is Steve Sumi's studies of that serotonin transporter polymorphism. And what I don't know so well is whether it has the kind of reproductive success kind of things that feel like they might have some evolutionary purchase, some natural selection purchase. I just don't know enough about the outcomes. I'm not sure, I don't know that he's particularly looked at, sort of reproductive success, for example, as a function of this. I can imagine, he can come up with a great intuitive story about how it would matter a lot given the social sensitivity of reproduction in macaques, but I just don't know empirically that it's actually happened. But that seems like, if anything, the most direct route are the genes that are involved in the behaviors and social competencies that structure reproductive success, even more so than immunologic success.